taken our sample from the field, and we don't have to be in a laboratory to do this, but we are in our UC Davis lab. So we need about a half a pound of sample like this, a composite of all of those different bales. And then I have that in this container, and I have a clearly marked bag, a Ziploc bag, and I want to take the entire sample of about a half a pound and keep that under seal. Particularly if you're interested in accurate dry matter, you have to keep it sealed. In fact, it would even be beneficial to double bag alfalfa hay samples, particularly if they're a little moister, so you don't lose some of that. We want to make sure it's clearly marked by the field, by the cut, and by the farm, so that the analysis can be performed by the laboratory correctly. Okay, so the concept is to have a sample that's large enough to represent the hay lot, but not so large that a laboratory is unwilling to grind a very, very large sample. And the whole point is that the entire sample should be ground by the lab. Generally, we see somewhere between 200 and 225 grams would be enough. That's about a half a pound. And so if you can bring in that amount, that's usually adequate for a laboratory to be able to handle. Okay, every sample has a certain number of stems per probe. Each stem typically represents a separate plant, or in some cases might be one plant. But in the case of GMO testing, we're looking for the population of stems that are in one probe and in one sample. And there are probably hundreds or if not thousands of stems in a single sample of the size about like this. And we've calculated that if you take a 12 to 14 inch probe, that you're likely to get about 250 to 270 stems represented in that probe. So therefore, depending upon the level of detection that you're looking for, you might be able to detect a low-level presence of a GE trait, an unwanted GE trait at a low level, with about 30 cores of sample. So after the sample is brought to the lab, it's ground, and for different types of analysis, sometimes only much less than this amount of sample is analyzed by the lab. So this represents the entire stack of hay. And so this is why it's so important to have a representative sample, even as it gets to this subsampling phase. When it's fully ground like this, subsampling is possible. That is, it represents the average if it's fully mixed, and if it's ground through a 1 mil type of grinder, then it can be representing the entire stack of hay. It's very important for the laboratory to grind the entire sample. That's why it's better from sampling point of view to have a very large sample, but the fact is a lot of labs will not grind the entire sample if it's 2 or 3 pounds worth of forage. But about a half a pound is probably practical for most labs to handle, and so a laboratory should be grinding the entire sample when it's brought into the lab. Once the sample is ground, it's very homogeneous in terms of leaf stem ratio. There are a few larger pieces, but most of the pieces are very, very small and can be fairly representative of the fiber as well as the protein and other characteristics.
So first we blow out all of the parts that the sample will be in to prevent cross-contamination. We take the powdered sample, and we pack it tightly. Blow off any excess. Then it's placed in the NAR. And we hit analyze. So right now it's scanning the reference standard right here. And now it says close the drawer. Now it's scanning the sample. And then we have data. And so it shows the dry matter content and what percent of the dry matter is protein and ADF, as well as other nutrients.